Hello, hi. Welcome back to a new lecture series actually, because we are in this lecture series we are going to deal about cellular energetics. Cellular energetic means the energy usage and the energy production in the cell. So we are going to study the exact mechanism of the reason of we consume food. What is the reason why we consume food? We know that food gives us something, right? Some energy and some important ingredient to our diet. So we will be studying what is the exact reason of we consuming food, how the food gives us energy. Okay, this is the chapter we are going to discuss in this your from your NCERT textbook, chapter number 12, respiration in plants. Look in the index, there's glycolysis, fermentation, aerobic respiration. Okay, we are going to discuss the three main things: glycolysis, fermentation, and aerobic respiration. So if you have noticed, the name of our chapter is respiration in plants. So does plants respire actually? What is actually respiration? In our case, human being, normal respiration, we are not talking about cellular respiration. Normal respiration means intake of oxygen, release of carbon dioxide. But plants, in case of plants, they also respire. They also release carbon dioxide. This is the respiration in plants. So we will be studying the exact mechanism. So plants also respire. They also release carbon dioxide. Okay. So the first process is glycolysis. And actually the respiration or the cellular respiration in every cell, whether it is plant, animal, or any cell, whether it is a unicellular organism, the respiration, we are dealing with the cellular respiration. So cellular respiration in any organism consists of a series of reaction, a set of reaction. The first reaction is glycolysis. Second is the TCA cycle, which is known as tricarboxylic acid cycle. Because why it is known as tricarboxylic acid cycle? Because the first product form is a tricarboxylic acid. It contains 3 carboxylic group. Okay, functional group COOH. It contains 3 COOH within the molecule. So it is known as tricarboxylic acid cycle. And then we will go to electron transport chains, complexes, and then to the fourth stage, which is the ATP synthesis stage. So this is actually the full steps involved in the energy production. I think you got an idea. We are going to deal about the energy production in a cell. Energy production in a cell start with glycolysis and end with the energy molecule, which is ATP, adenosine triphosphate. So energy in cell is produced in the form of ATP. Why it is produced in the form of ATP rather than direct production of energy, we will be dealing here. What is the use of certain large processes? If you think the energy can be made in single step and energy can be made in multiple steps, but why an organism, its mechanism have evolved in such a way that it happens, the energy production steps happen in a multiples of steps? Yes, glycolysis, TCA, and next the electron transport, next the ATP. Even in glycolysis there are 10 steps. So why? What is the reason for the production of many steps? It can simply get converted, but cell does not convert it. Look, in our automobiles, which runs on the fuel hydrocarbon, which runs on hydrocarbon fuel, if you notice it, The energy is derived from the hydrocarbon molecule, like octane, right? But most of the energy is partially oxidized, wasted, not fully oxidized, not fully combusted. And most of the heat energy is released in the form of heat. That means most of the energy from the fuel is lost as a form of heat or as a form of other chemicals, partially oxidized chemicals. So, but in case of organism can't afford any such waste process. So what does an organism do? The organism, from the course of evolution, it has evolved a much more efficient mechanism so that the wastage of fuel is very low. I think you are getting some basic idea of it. What we are studying in this lecture, we are studying about how a cell makes its energy, how glycolysis provide you energy to study or understand glycolysis. What is the purpose of eating food? These are the things we are going to study. I will be talking in a general manner. Then we will come to the respiration in plants. Respiration in plants means plants also breathe by releasing carbon dioxide. That is the thing. Plants utilize oxygen and release carbon dioxide. This is known as a respiration in plants. But what happens in photosynthesis? What is the purpose of photosynthesis? Photosynthesis is the production of the energy molecule. Sorry, a carbohydrate, right? So photosynthesis require carbon dioxide, it require water, it requires sunlight. These are the basic things for photosynthesis, and within this they will be making a carbohydrate source. The carbohydrate source here the plants make is glucose. The same glucose is required for glycolysis. What is glycolysis? Glycolysis means breakdown. Lysis means breakdown of glucose. Glyco means sweet, actually. Lysis means breakdown. Okay. Actually, glucose is a carbohydrate. CN having a molecular formula CnH2NON. Right? This is the general equation for a carbohydrate. What are carbohydrates? They are polyhydroxy ketones or polyhydroxy aldehydes, or the substance which yield these things on hydrolysis. This is actually carbohydrate. So plants make carbohydrate, especially glucose. The photosynthetic product is glucose. So that is photosynthesis. But plants utilize this product, which is the photosynthetic product glucose, for various of purpose. First is the produced glucose is used as their energy source when the glucose enter into the process of glycolysis. Before going to glycolysis, I am saying some important points so that you can make a general idea of what we are going to study. Okay, I am not directly going, because you need to understand glycolysis if you are continuing to study biology in your future also. This is the main thing. Energy is needed for every cellular processes. Energy is released or made in the form of ATP, adenosine triphosphate, mainly ATP. Some other molecules are also there, GTP, etc. But ATP is the main energy currency in the cell. ATP is the energy currency in the cell, and mitochondria is the powerhouse of the cell. We will deal why they are known as powerhouse, because the glycolysis, everything, this respiration occur in the mitochondria. Glycolysis occur in the cytosol, the fluid, and the rest process occur inside the mitochondria, and the ATP is produced inside the mitochondria. So it is known as powerhouse of the cell because it produces energy molecules. If you remember, while we were discussing the sodium potassium pump, I mentioned sodium potassium pump is a P-type pump, right? If you remember, P-type pump means it needs phosphorylation. The pump, what is a pump? Pump is a thing which needs energy, right? So energy is given by ATP by giving its phosphate to the pump, so the pump get activated. This is the one use of ATP. There are various uses of ATP inside the cell. I'm just giving a small example of how this ATP molecule is used, why ATP molecule is used as a energy currency in the cell. This is a small example. So the ATP is produced in the process of a series of set of reaction. The first reaction is glycolysis, which occur in the cytosol. Cytosol means it's fluid in the cytoplasm. And then after glycolysis, a product is formed. This product is transported into the mitochondria. Then the next three steps, which is the TCA cycle, then the electron transport, then ATP. This is synthesized inside the mitochondria. So mitochondria is known as the powerhouse of the cell. Actually, if you don't want to hear these stories, you can skip. Actually, why I am saying this is because to make a general understanding of the topic, so that it will be useful for your future also, because this is a topic which is needed till the end of your biology career. Okay. So in photosynthesis, plants produce glucose. Okay, this glucose is used in a variety of processes in plants. One of the processes is the usage of the glucose as a storage form, in storage form. So glucose is a carbohydrate. We know that glucose is easily soluble in water. Carbohydrates are organic compounds. We know that organic compounds are insoluble in hydrophilic solvents, right? But exceptions are seen. The exception is the carbohydrates, which contains more oxygen bonds, so they are easily soluble in water. And due to this reason, glucose can't be stored in as a glucose form. So glucose is converted into a big molecule, a polysaccharide. Polysaccharide means so many monosaccharides joined together by glycosidic bond. Okay, so so they form a molecule which is known as starch. So in plants, the storage polysaccharide is starch. Starch is a homopolymer of glucose linked by alpha 1,4 linkage, which means one glucose and the other glucose is linked by alpha 1,4 linkage. First and the fourth position of the first and the second molecule, second glucose. Okay, so the plants store glucose in the form of starch because glucose is easily soluble in water and get solubilized, so it can't be used for any other purpose. This is the first thing when glucose is formed by the process of photosynthesis. Next, the glucose is transported as sucrose. Sucrose, glucose transport it in the form of sucrose. Sucrose is a disaccharide, contains fructose and glucose. So why it is converted into a glucose? Why it is converted into sucrose? Because it can be easily transported to all parts of the plant. This is the other thing. So it is stored as starch. It is also converted to sucrose. Next, it is converted into cellulose. Cellulose is a structural polysaccharide which is used to make the structure of the plant cell. Okay, so cellulose is also a form of glucose. It is also a polysaccharide of glucose, and it is a structural polysaccharide of glucose, which means it is used to make the structure of the cell. And the linkage is beta 1,4 linkage. Just a general idea. Glucose is made by the process of photosynthesis, and these are the uses of the glucose. And the last usage of the glucose is that the glucose is used to power their cell. How? By the process of glycolysis. I mentioned what is glycolysis. Glycolysis, breakdown of glucose. Okay, a very easy reaction. Okay. So first we will be studying. There are some general definitions. Actually, the students who are preparing for the NEET does not need to study any definition. They need just to understand the concept. So what is cellular respiration? or it is the mechanism of breakdown of food materials within the cell to release energy. Yes. Okay. So there was a question arising in your textbook whether plants breathe. Yes, plants require oxygen for respiration. Yes, by the process of cellular respiration, they need oxygen. So what actually happens in the cellular respiration? Cellular respiration, it is also seen in every organism. So it is a general theme, not only in plants but also in every organism. If you look at the equation C6H12O6, it is an equation for a carbohydrate containing 6 carbon, which is glucose. We are considering it as glucose because glucose is having 6 carbon, C6H12O6. Glucose is also having its isomeric forms, which is mannose and galactose. It is also having isomer form, which is the fructose. Okay, so glucose, by the process of the photosynthesis, glucose is produced. This glucose is now entering the cellular respiration. So glucose, with the help of oxygen, that converted into energy, and the waste products are carbon dioxide and water. They are released in the cell. Only energy is required, right? The rest is waste. Same as that in case of photosynthesis, where oxygen is a waste product. Okay, we will study photosynthesis in the coming. After this is completed, we will now go to the photosynthesis. This is the basic idea. Actually, you need to read your textbook for basic understanding. First, you should understand the class, then go for your textbook. It will be difficult if you, some people may feel difficult if they first go and read the textbook. So it is better you make some basic understanding about the chapter and then go to the text. Okay, these are some definition. Okay, now I'm directly going to the process of glycolysis, then we can come back to the definitions. Okay, this is glycolysis. Look, okay, I am saying once more, glycolysis is the breakdown of glucose to pyruvate. Okay, then the pyruvate will be converted into acetyl coenzyme A. Then it will be converted, then it will enter TCA cycle. From TCA cycle, some substance will be produced. These things need to be, they will be entering electron chain, transport chain, and then they will enter ATP, and they will be an ATP will be produced. I earlier mentioned it is a set of processes. It is not a single reaction. It is a set of reaction. Then only the last ATP will be produced. Okay. So, glycolysis. Glycolysis contains 10 steps. Glycolysis contains 10 steps. How to study glycolysis is that first you need to understand the steps. Look, there are look, if you count it, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, 10 steps which have 10 substrates within it. Okay, 10 intermediates or 10 substrates within it. 10 molecules are involved. 10 steps. So this is the thing first I mentioned, why glucose can be directly converted into pyruvate or pyruvic acid. If glucose is directly converted into pyruvate, then most of the energy will be waste as heat. So the glucose fuel or the carbohydrate fuel can't be utilized successfully. This is the reason why cells choose glycolytic pathway, and this pathway contains 10 steps so that they can maximize the energy present in the bonds. Okay, I think you are getting some understanding. I will make you perfect after this, but just follow up. It will be little bit difficult for the students who are learning it for the first time. Okay, so first is glucose. Okay, the thing to understand is that how to study glycolysis. first you need to understand what are the substrates formed. 10 substrates are there. So we need to understand the names of, you need to study the 10 substrates names. Then only we can write, you can make a basic understanding, right? So you need to first memorize. I will say memorize because it is needed. If you read it only 2-3 times, it will be easy for you, but you need to understand. So the first molecule is glucose. Glucose then convert. So I'm just saying glucose that convert any glucose 6 phosphate, then fructose 6 phosphate, then fructose 1,6 bisphosphate. So then fructose 1,6 bisphosphate is converted into two molecules, glyceraldehyde 3 phosphate and DHAP, which means dihydroxyacetone phosphate. Okay. Then these compounds are actually isomers. So they can be returned back to glyceraldehyde and it will form 1,3-BPG, 1,3-bisphosphoglycerate, 3-bisphosphoglycerate, 2-bisphosphoglycerate, phosphoenol pyruvate, and pyruvate or pyruvic acid. So the how to study glycolysis is that you need to first by heart the names of the intermediates. That is the only way. Then if you by heart it, it will be easy. So what I am going to do here is that first I will say the names, then I will say how glucose is being converted into the intermediates by understanding, making you understanding with the structures. Okay. So let's start. I think you got some basic idea. Let us start. The first step is the conversion of glucose into glucose 6 phosphate. Okay, just understand glucose to glucose 6 phosphate. I'm just going to draw the structure. Okay, glucose to glucose 6 phosphate. What is glucose? Glucose is a 6 carbon carbohydrate, right? I think you know structure of glucose. C 1 2 3 4 5 6. This is a 6 carbon glucose glucose backbone. So I'm going to write CHO CH2OH. Okay, then in third carbon OH will be oriented towards the left. The rest of the OH will be oriented towards the right. This is the structure of glucose. Okay, it may be a little bit boring for you because why I am drawing this structure, it is to make you understand, or else you can just by heart it. By heart means by heart the entire process, but it will be difficult for you in the future, in your future. So that is the reason I am saying with the structures, so that you will get a good understanding what happens, why this is happening, why this is not happening. So actually it will be a lengthy lecture today. I think you should be patient. You should have patience within you because it is an important concept in biology. Everywhere you need energy. So this is the structure of glucose. I'm just writing G or glucose. Okay. So this is the structure of glucose. The most common structure of the glucose is this. Glucose is also having many structure, which means DL form. I'm drawing here the D form, which is the most common form which is seen in the organism. So what was the first step? The first step was the conversion of glucose into glucose 6 phosphate. Okay, now I am drawing the structure of glucose 6 phosphate. The structure will be same here till here, and the sixth carbon, which is CH2O P. P means a phosphate group is there. You should write the structures till here, which means CHO, C, 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 and CH2OP. Okay, you should write it. I am not drawing here. Look, this is known as glucose 6 phosphate. What is glucose 6 phosphate? First step is conversion of glucose into glucose 6 phosphate. So what is the change here? A phosphate is being added, right? What? A phosphate is being added in the 6th position. We need to understand that in this structure, in this glucose, only the phosphate can be added very easily in the 6th carbon because of the bonding. Because of the bonding. I'm not going much deep, just understand because of the bonding. Phosphate can be easily added to this position, the 6th position. So it will become glucose 6 phosphate. Okay. So who gave phosphate? That is a question, right? The phosphate is given by ATP. ATP gives one phosphate. Thus, if ATP gives one phosphate, it get converted into ADP, adenosine diphosphate, because adenosine triphosphate, one phosphate is loosed, it will be forming adenosine triphosphate. This is the first step. And glucose to glucose 6 phosphate. Okay. So, who gave the phosphate? The phosphate is donated by ATP. Look, now ATP came, right? So, ATP is this is one of the other role in ATP. For the usage of ATP, it is used to power some cellular machineries. This is one of the example of the use of ATP. So you will be thinking, we are going to make ATP. We are going to make ATP with the use of ATP, right? Yes, this is correct. We are going to produce ATP with the use of, first we will be starting the reaction with ATP. Like burning wood. The wood will not burn as it is, but if we ignite it with some energy, we are making the delta G negative, then the reaction will be spontaneous after ignition. Not spontaneous, the reaction will continue. So it needs some initial ignition. The initial ignition here is ATP. We use some ATP to produce ATP. So glucose 6 phosphate is produced. So there is an enzyme which add or which breaks this phosphate and add this phosphate to the glucose, which is a kinase. Kinase enzyme means the enzyme which add phosphate. So here is it is a kinase, and the name of the kinase is hexokinase. Hexo means 6, right? Hexokinase. The name is given according to the structure. You should not ask why it is given. It is given because of the structure. Hexokinase. So kinase means the enzyme which add potassium. So potassium. So the name of the enzyme is hexokinase. So glucose have been converted into glucose 6 phosphate with the help of ATP and with the help of a kinase. The name of the kinase is hexokinase. This is the first step. Okay. Next, what was the step? Conversion of glucose into glucose 6 phosphate into fructose 6 phosphate. Conversion of glucose 6 phosphate into fructose 6 phosphate. So we need to understand the structure of fructose. I will be drawing fructose is a isomer of glucose. Okay, so fructose 1, 2, 3, 6 carbon, right? 4, 5, and 6. Fructose is a 6 carbon isomer of glucose. CH2OH. I am drawing the structure. CH2OH. Here 2 CH2OH. Actually, it is a lengthy step, lengthy video. If you want to skip, you can skip. I am just saying the structure. After this, I will just say the overall idea. If you don't want to hear this, you can skip. But it is very important that if you hear, it will be better or beneficial for your future studies. Okay. So fructose structure. Fructose, we know that fructose is an isomer of glucose. Glucose is a aldose form, and fructose is a keto form. So keto, C double bond. All keto form will be having double bond O in the second carbon atom. This is the keto form. Okay. So this is fructose. Fructose 6-phosphate. We have not drawn the phosphate. I will rub it and draw the phosphate here. Actually, I draw for the structure of fructose. So a phosphate group here. Phosphate. Phosphate means actually it is phosphoric acid. Okay, not phosphor added. Okay, it is PO3 2 minus. PO3 2 minus is being added because ATP is having 3 PO3 2 minus. So one of the PO3 2 minus has been cleaved by exokinase and it has been added to glucose 6-phosphate. The next step is just a conversion of the glucose to its fructose, its isomer form. So an isomerase enzyme is actually needed. An isomerase enzyme is actually needed. These are isomers, so they can easily exist as in equilibrium form. Okay, so okay, this is the structure of fructose. The rest of the OH is oriented towards the right. This is the structure of fructose. I think you got an idea. So we just completed the 3 steps. Now only 7 steps are left. So glucose to glucose 6-phosphate and glucose 6-phosphate to fructose 6-phosphate. Okay, here the first ATP is used in the first step. I said why it is used, because an energetically unfavorable reaction, we made it favorable by using ATP hydrolysis. This is the purpose of these 10 steps glycolysis, because the first molecules in the glycolytic pathway are very high energy molecules, so that each molecules converted we can easily utilize the energy, easily utilize the bond energy to make the ATP. Okay, so an isomerase enzyme is used here. Isomerase enzyme. An isomerase enzyme. Isomerase enzyme. What does an isomerase enzyme do? It just convert one form of an molecule to its isomer. Keto form into aldose form, aldose form into ketose form. Here the isomer's name is phosphofructoisomerase. Phosphofructoisomerase. Actually, for NEET students, the name is actually not needed, but I'm saying phosphofructoisomerase. Phosphofructoisomerase means the name is given according to any one of the molecule, which means fructose can be converted into glucose, glucose can be converted into fructose. So the name has been given as phosphofructoisomerase. Okay. So we have completed the 3 steps. Now, what happens in the next step? Fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Okay, I will just show. Look, glucose converted into glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate. Next, fructose 6-phosphate into fructose 1,6-bisphosphate. The one thing to remember is that these structures will be in its cyclic form. A ring structure. Usually, a ring structure will be formed here. But I am not drawing because it will be very, very confusing for you if I am drawing the ring structures of the molecules. So I am not drawing the ring structures. I am just drawing the general structure or the linear structure, so you can understand it better. Okay. Next is the conversion from fructose 6-phosphate to fructose 1,6-bisphosphate. I'm just rubbing it. I'm sure you have written it into your notebook. Okay. Next is the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. What do you mean by 1,6-bisphosphate? Means what was fructose 6-phosphate? Fructose 6-phosphate means a phosphate group in 6th position. Now it got converted into fructose 1,6 bisphosphate, which means first position and sixth position of the fructose contains phosphate group, okay, or phosphoric acid group. Okay, so we can draw the structure. See, actually I will draw here. I think you have written it, right? So it just converted into fructose 1,6 bisphosphate. You just understand this is fructose 6 phosphate. We are going to convert it into fructose 1,6 bisphosphate, means here one phosphate is being added, phosphate group has been added. So it became fructose 1,6 bisphosphate. So what made it fructose 1,6 bisphosphate from fructose 6 phosphate? By the addition of a phosphate group. So here comes our next ATP molecule. So an ATP molecule got broken down and it releases phosphate group. ATP got broken down into ATP and the phosphate group is being added to the first carbon. So it became fructose 1,6 bisphosphate. So this phosphate group has been added by an enzyme. So it will be a kinase enzyme. Okay, so a phosphate group has been added. So fructose 6 phosphate get converted into fructose 1,6 bisphosphate and with the help of an ATP. Okay, so ATP got broken down into ADP and the phosphate has been added and a kinase enzyme is used. Okay. Okay. Next. So this is the fourth step. Yes, this is the fourth step. Next, the fifth step is the conversion of, okay, now fructose 1,6 bisphosphate is formed. Now it is the crucial step we need to understand. Fructose 6 phosphate has been formed. Now the fructose 6 phosphate get broken down into half, which means fructose is a 6 carbon molecule. Now it will get broken down into three compounds, which means 3 carbon compound and 3 carbon compound. So the first 3 carbon compound is glyceraldehyde 3 phosphate and second 3 carbon is DHAP, dihydroxyacetone phosphate. Look, from the name itself we can understand glyceraldehyde 3 phosphate. Third position of the glyceraldehyde contains a phosphate. We know that there are 2 phosphate. So one phosphate is gone with one of the molecule, one of the 3 carbon molecule. Next, the 3 carbon molecule is the dihydroxyacetone phosphate, DHAP. So it is also having a phosphate group within it. So first I will draw glyceraldehyde 3 phosphate. It is an aldose form of the 3 carbon molecule. Aldose form of a sugar. It's a 3 carbon. 1, 2, 3. I am going to draw glyceraldehyde 3 phosphate. It is an aldehyde group. So CHO, CH2, OP. Okay. Actually, if the phosphate was not there, it will be CH2OH, which is glyceraldehyde. Okay. OH. H. This is glyceraldehyde 3 phosphate, and the next compound is DHAP, dihydroxyacetone phosphate. It is a keto form of a 3 carbon sugar. So keto form, C. I mentioned that all keto form of the sugar will have the second carbon atom O, having containing a double bond O, because it is a ketone, RCOR. That is the structure of a ketone, right? So CH2OH, what I said, dihydroxyacetone phosphate. So here CH2OP. Okay, this is DHAP, dihydroxyacetone phosphate. So they both are isomers. Yes, whether they are isomers? Yes, they are isomers. This is the aldose form, this is the ketose form. They are 3 carbon molecule. Yes, they are isomers. So they exist in an isomeric form. So I will write they exist in an equilibrium form, equilibrium, and they can be interconverted by the help of an enzyme which is known as TIM. TIM means triisomerase enzyme. Triisomerase enzyme. Okay. So it is an isomerase enzyme TIM. Okay. So fructose 1,6-bisphosphate got converted into 3,3 carbon molecule, and they also exist in isomer. They also exist as they also exist in equilibrium. They also exist in equilibrium with each other. So it is converted with the help of an enzyme aldolase, which made it as aldose and ketose form. So it is by the enzyme aldolase. Okay. Now we got into the next step. So this is the condition. I think you got an idea. So if you have thought, okay, 1 minute. Look, glucose 6-phosphate got converted into fructose 6-phosphate, then fructose 1,6-phosphate. Then if you think you should may got a doubt that why glucose didn't convert it directly into glucose 1,6-phosphate, and thus thus to the next step. I think I will say it once more. Why glucose change into fructose and then fructose 1,6-phosphate? Why not glucose 1,6-phosphate? This is my question. I will say it once more. Why did fructose 1,6-bisphosphate changed into the next step rather than glucose 1,6-phosphate? What is the reason for the conversion of glucose 6-phosphate into fructose 6-phosphate and then fructose 1,6-phosphate? Because there is a good reason for it. Because if glucose 6-phosphate has been converted into glucose 1,6-phosphate and the next step, then we will not be getting 3 carbon compounds. We will be getting a 2 carbon compound and a 4 carbon compound. I will say, oh sorry, I have erased it. Look, just consider it as glucose 1,6-bisphosphate. If it was glucose 1,6-bisphosphate, just it is actually fructose, just consider it as glucose 6-phosphate, glucose. So if this was glucose 1,6-phosphate, the aldolase enzyme will cleave here. If the aldolase enzyme cleave here, then we will get a 2 carbon compound and a 4 carbon compound. 2 carbon compound and a 4 carbon compound. But what actually is happening is that if fructose 1,6-bisphosphate is present, the aldolase can't cleave in the second position. It can only cleave in third position. So we will be getting a 3 carbon compound. Actually, normally aldolase cleaves here in this bond. Okay. So we will be getting 3 carbon compounds. But if glucose 1,6-bisphosphate is used, the aldolase can't cleave it equally. So we will be getting a 2 carbon compound and a 4 carbon compound. So what will be the after effect? The after effect, we need separate pathway for a 2 carbon compound and a 4 carbon compound. But look here, DHAP and glyceraldehyde 3-phosphate are formed. Both of them are 3 carbon compounds. So we need only a single pathway to continue. But if it was 2 carbon compound and a 4 carbon compound, we need different enzymatic machinery. We need different pathway. So it is a waste of energy. So this is the reason why glucose 6-phosphate just converted into fructose 6-phosphate, then fructose 1,6-bisphosphate. Okay, I think you got an idea regarding it. So this is the step. Now we can go to the next step. So we reached here, right? Now we are going to the next step. Okay, I will just rub it. Okay, next I said that these both compounds are interconvertible. This is known as glyceraldehyde 3-phosphate. So even if the DHAP is formed, it will be getting converted into glyceraldehyde 3-phosphate. The next pathway is from glyceraldehyde 3-phosphate. Okay. Okay. Because these DHAPs are required in the next in most of the reactions. So glyceraldehyde 3-phosphate is being used. Next, glyceraldehyde 3-phosphate get converted into 1,3-BPG. What? 1,3-BPG. 1,3-BPG means 1,3-bisphosphoglycerate. Glycerate. It was a glyceraldehyde, which means CHO. Now the CHO is being converted into glycerate or glyceric acid. You know why it is known as glycerate and why it is known, it can be also called glyceric acid. Look, I will just say an example, a small example. You know the structure of acetic acid, right? CH3COH. CH3COH is known as acetic acid. This is known as acetic acid, and if they exist in a ionic form, usually in the physiological pH of the cell, these compounds will be existing in an ionic form. So this is known as acetate. So what is this known as? 1,3-BPG is known as 1,3-bisphosphoglycerate or 1,3-BPG glyceric acid. Okay. Look, I will just say it once more. From the beginning, glucose to glucose 6-phosphate to fructose 6-phosphate, fructose 6-phosphate to from fructose 1,6-bisphosphate to the fructose 1,6-bisphosphate, which is a 6-carbon compound, is cleaved by the help of an aldolase enzyme into 2 3-carbon compounds that we have discussed, which is glyceraldehyde 3-phosphate and DHAP. Next step is the formation of 1,3-BPG. 1,3-BPG. So what is 1,3-BPG? 1,3-bisphosphoglyceric acid or glycerate. I just mentioned how it is called as, why it is called as glycerate, because in physiological pH they exist as ion. Okay. Now we will draw the structure of 1,3-BPG. 1,3-BPG. You know the structure of glycerol. From the structure of glycerol, 1,3-BPG is derived. So first let us draw the structure of glycerol. Glycerol CH2OH 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 and CH2OH. This is the structure of glycerol. In case of 1,3-BPG, it is a glyceric acid. So glycerol, when it is oxidized, it can be turned into glyceraldehyde, which means CHO. This is the thing. Now further oxidation can create glyceric acid, which means COOH. This is known as glyceric acid or glycerate if it exists in the ionic form. So what happens in case of 1,3-BPG? The structure of 1,3-BPG is first and third carbon will have a phosphate group within it. So OP. Okay. This is the structure of 1,3-BPG, 1,3-bisphosphoglyceric acid or glycerate. It is actually formed from glyceraldehyde 3-phosphate. DHAP is interconvertible, or it exists in equilibrium, so it convert back into glyceraldehyde 3-phosphate, and from glyceraldehyde it is converted into glyceric acid. So how an aldehyde is converted into an acid? And you see that an extra P is being added. Here there was only the presence of one phosphate group which was in third position, but when we convert it into 1,3-BPG, they have the presence of an extra phosphate group in the first position. Okay. So how it has been formed? It is with the help of a, so if we look to the structure of glyceraldehyde 1,3-BP G3P, glyceraldehyde 3-phosphate, we can understand that here it is aldehyde. Okay, an extra O, look, an extra O is being added here, and extra P also. If you analyze both the structures, from this structure this has been formed, so an extra O with an extra P is formed. So from where does this came? From where did they came? So it is actually donated with the help of phosphoric acid. Okay, I will actually once more draw the structure very neatly, so you should write this in your notebook. You should also remember that they exist in equilibrium. I will draw once more the structure of 1,3-BPG. Okay. It is a 3 carbon, 1, 2, 3, CH2, OP, here COOP and CHOH. Okay, so my question is that from an acid aldehyde, an acid is formed. Also, this acid have the presence of a phosphate group within it. So how has it been formed? It is, I said, it is with the help of a phosphoric acid. Phosphoric acid donated the P group, also the oxygen. Oxygen is from the phosphoric acid. So I will say how did it happen? First of all, I will say structure of phosphoric acid, H3PO4. It is the phosphate donor in ATP also in this case. So phosphorus is the 15th element in the periodic table. So electronic configuration will be 285, which means the valence electrons contain 5 electrons. So I will draw the structure 1, 2, 3, 4 and 5. This is held with oxygen. The rest of it are having hydroxyl group within it. This is the structure of phosphoric acid. Okay, this is the structure of phosphoric acid. So phosphoric acid react with the glyceraldehyde 3-phosphate. What happens is that this H from the glyceraldehyde, also this H from the phosphate group is released within the process, and it is accepted by nicotinamide, NAD. It is accepted by NAD, nicotinamide. So 2 hydrogen has been released from here and here. So that 2 hydrogen is accepted by nicotinamide, NAD, nicotinamide dinucleotide, and it get reduced to form NADH2, or we can write NADH, H plus. Okay, so this is the condition. I know it is little bit boring for you because it is biochemistry part, but if you understand this, you can understand glycolysis very easily. So nicotinamide NAD accepts and forms NADH2. It undergo reduction, and the phosphate group is donated by this phosphate is donated by H3PO4 or phosphoric acid. So this P is actually the entire structure without H. That is the thing I said, that this O is the O donated by the phosphoric acid. Okay, so now 1,3-BPG have formed. So the enzyme here is 1,3-BPG dehydrogenase. Okay. Yes, sorry. Oh no, no. Glyceraldehyde 3-phosphate dehydrogenase, because the enzyme here is glyceraldehyde 3-phosphate dehydrogenase, because a hydrogen has been released. So the enzyme here is glyceraldehyde 3-phosphate dehydrogenase. Enzyme is actually a dehydrogenase enzyme. Okay. So now what? This is the condition. We have reached here 1,3-BPG. Now the next structure is 3-BPG. 1,3-BPG to 3-BPG. So I will wrap the entire thing. I am saying it once more. The structures are not needed for you, but if you study the structure, it will be best. Okay, so we have formed 1,3-BPG. Now from 1,3-BPG, the next structure is this also exists in an equilibrium. The next structure is 3-phosphoglycerate, right? 3-phosphoglycerate or 3-phosphoglyceric acid. I said why it is known as glycerate or glyceric acid. So what happens in 3-BPG? One of the phosphate is ready. One of the phosphate is removed. Okay, so it becomes H. So what happens? The reaction is one of the phosphate is removed, and the removed. So what happens from 1,3-BPG? It is converted into 3-BPG. In 3-BPG, what is the difference between 1,3-BPG and 3-BPG? BPG means phosphoglycerate or glyceric acid. The difference is that one phosphate is removed. So the removed phosphate is accepted by ADP to form ATP. Look, now the role of the glycolysis comes, because a phosphate has been, an ATP has been formed. So ADP accepts one phosphate removed from 1,3-BPG and it forms ATP. So here the first substrate level phosphorylation happens. Phosphorylation means addition of a phosphate group. So this is the first ATP product producing step. This is known as substrate level phosphorylation. Then what was the first reductant step? The first reductant step was 1,3 glyceraldehyde 3-phosphate to 1,3-BPG. It was the first reductant producing step. Yes, NADH2 was formed. It was a reductant. I said how it is formed. So throughout this reaction, throughout the glycolysis, we will be getting ATP. ATP will be formed. NADH2 will be formed. So you will be having a common doubt. Why are these formed? Because these formed molecules will have a greater role in the upcoming reaction. In glycolysis, we got ATP. So cell got ATP, but we need more ATP. So these more ATP are seen in the NADH2 as a high energy molecule. They are high energy molecules because they, in the coming third step and fourth step, can produce ATP. We will see how NADH2 leads to ATP production. So they are also important molecules. They are not waste products. NADH2 is formed because they have a certain role in the upcoming reaction, even though they do not have a role, but they have an indirect role in glycolysis and gluconeogenesis. We will study in the coming part. So actually, if you don't want to hear these things, you can skip, and after that I will say you can skip and check which is the part. That part I will be just briefing the pathway, but actually this pathway is important to understand. If you understand the structure, this will be very beneficial. So we have formed from 1,3-BPG, we have came to 3-BPG, and from 3-BPG, so look here, an ATP is formed from 1,3-BPG to 3-BPG. ATP is formed. From 3-BPG, there is also a step to form 2-BPG. 2-BPG. which means this phosphate is just interchanged with the second carbon. So it is with the help of a mutase enzyme. Mutase, just write mutase. Mutase enzymes helps in shifting the functional groups within the carbon molecule. So from 3-BPG, 2-BPG has been formed, and the enzyme here is phosphoglycerate kinase. Phosphoglycerate kinase here. We know that kinase is an enzyme which add phosphate group. So in the from 1,3-BPG to 3-BPG, a kinase enzyme is added. The name of the enzyme is phosphoglycerate kinase. Means it is adding a phosphate. Just if you are having confusion in the name, just consider the reaction from 3-BPG to 1,3-BPG. So there 3-BPG to 1,3-BPG, a phosphate is added. So the kinase enzyme, the name is given in the reverse order. Only that is the thing. Because while these reactions were conducted, the scientists have given the name of the enzyme on the basis of a particular reaction, any reaction, because these exist in the equilibrium form. So scientists gave the name when the molecule when the reaction is from 3-BPG to 1,3-BPG. If the molecule reaction is from 3-BPG to 1,3-BPG, which means a kinase has been added, a phosphate group has been added, and it is by the help of a kinase enzyme. So phosphoglycerate kinase. So from 3-BPG to 2-BPG, 2-BPG means this is the structure, second carbon will be having phosphate group. Now 2-BPG to phosphoenol pyruvate, the second last step of our reaction. What? Phosphoenol pyruvate. Phosphoenol pyruvate. I will draw the structure once more. So 3-BPG to phosphoenol pyruvate. 3-BPG to phosphoenol pyruvate. Let's draw the structure of 2-BPG once more. 1, 2, 3. It is the structure of glyceric acid. 2-BPG means CH2OP. CHOP. Okay. From here, we are forming phosphoenol pyruvate or phosphoenol pyruvic acid, which means double bond CH2 . First we will draw the structure of pyruvate. What is the structure of pyruvate? The structure of pyruvate is this. This is the structure of pyruvate. This is the structure of pyruvate or pyruvic acid. Now phosphoenol pyruvate means OP. This is the structure of phosphoenol pyruvate, the second last substrate. The second last substrate is the phosphoenol pyruvate. So this is 2-BPG. 2-BPG get converted into phosphoenol pyruvate, the second last structure. This is the structure phosphoenol pyruvate. So what is the difference between 2-BPG and phosphoenol pyruvate? Here okay. So what is the difference between 3-BPG? Actually it is CH2OH. CHOH. Okay. 1, 2, 3. Okay. So what is the difference between 2-BPG and phosphoenol pyruvate? If you look into it, we can see that H2O has been released, right? Because it is the enol form. So an enolase enzyme will come. The enzyme here is enolase enzyme, and the difference is that a double bond has been formed and what H2O has been released. Just check, H2O has been released. There's CH2OH here. Let me draw once more. CHHOH. So CH2OH. From CH2OH, it has been converted into phosphoenol pyruvate, enol form. So an enolase enzyme helps it and water molecule is released. Yes, please check it because one hydrogen, one oxygen, one hydrogen is released. So H2O is released. This is the condition of phosphoenol pyruvate. Next, the last step, this also exists in equilibrium. So the last step is the formation of pyruvate from phosphoenol pyruvate. This is the structure of pyruvate or pyruvic acid. So what happens? So a phosphate is released. Yes, the difference is the phosphate is released. If a phosphate is released, it will be accepted by ADP forming ATP. So the second level phosphorylation happens, substrate level phosphorylation happens here, which means another ATP is formed. Look, the result of glycolysis, we got 2 ATP, one in this step and the previous step, which means 1 3-BPG to 3-BPG. There are also ATP form, which was the first substrate level phosphorylation. Here the second substrate level phosphorylation also happens. So it will be also with the help of it is done with the help of a kinase enzyme. Actually, kinase is the addition of the enzyme which add phosphate group. Yes, it is adding. Just check the reverse. Just check the reverse condition. From pyruvate, this is pyruvate, phosphoenol pyruvate, which means the phosphor group is added. So the enzyme is kinase enzyme. Actually, the name is given. The name of the enzyme is given in the reverse order. Okay, so this is the condition. So we ended up the glycolysis process. Now this is the end of the glycolysis. Now I will say it once more. What happens in glycolysis? The glucose is converted into pyruvate. This is the thing. But the conversion happens in a 10 steps. 10 steps. Why 10 steps? The 10 step is to maximize the utilization of the energy inside the bond. So what happened? We first gave 2 ATP and the result was 4 ATP. Okay, we first gave 2 ATP which was used for the initiation of the process and we got 4 ATP. 4 ATP. I will say how the calculation is done. Actually, 1 ATP is used and we got 2 ATP back. But why did I say we got 4 ATP and we used 2 ATP? Because there is a thing you need to understand. Look, so I think you got a basic idea of the glycolysis. Now we are going to the calculation of the ATP. Glucose was a 6-carbon molecule, but the 6-carbon glucose got splitted into 3-carbon in this step, right? If you remember, in this step it got splitted with the help of a aldolase enzyme. It got splitted into 3-carbon. So there is actually 2 parallel pathways going on, which means if this molecule is converted into this triose or glyceraldehyde 3-phosphate, actually there are 2 molecules. That means 6-carbon molecule glucose is converted into 3-carbon molecule pyruvate. So according to the basic logical mathematics, if one 6-carbon glucose can be converted into 3-carbon pyruvate, then 2 pyruvate can be formed. That means one glucose can give 3 carbon. One glucose can give 2 pyruvate. One glucose can give 2 pyruvate. This is the result of glycolysis. One 6-carbon glucose can give 2 3-carbon pyruvate molecule. So let us calculate the ATP. ATP usage and ATP produced in the glycolysis. So 2 ATP has been used. I will write the net result of glycolysis. Look, 2 ATP has been used, right? 2 ATP has been used. And which were the steps using 2-ATP? This was the step and this was the step which uses 2-ATP. Now, ATP was produced also in 2 steps, which is this step from 1,3-BPG to 3-BPG, also from phosphoenol pyruvate to pyruvic acid. But here the condition is different. Here it is 2 times. Why 2 times? Because the 6-carbon molecule has been split into 3-carbon compounds. The 3-BPG, which is actually 2. So, two ATP formed here, 2 ATP formed here. So actually 4 ATP formed. 4 ATP formed. I think you are understanding. It is little bit confusing for you. I think so. Actually 2 ATP is used first here. 1 ATP here, 1 ATP here used. And 4 ATP is formed. How 4 ATP is formed? Because it is 3 carbon, 2 molecules of 3 carbon. So here actually 2 ATP is used. Here also 2 ATP is used, because from this reaction onwards it is 3 carbon molecules. Okay, so 2 ATP used and 4 ATP produced. So what is the net result? The net result only 2 ATP gained, because 4 minus 2. From the glycolysis reaction, for converting 1 glucose into 2 pyruvate molecules, we gave 2 ATP. We got 4 ATP. The net is actually 2 ATP, because 2 has been used for the initiation of the glycolysis. And we got also reductant molecule. 2 reductant molecule has also been formed. With 4 ATP, 2 NADH2 has also been formed. What is the use of NADH2? NADH2 is also used to produce ATP in an indirect way, which we will be studying in the stage 3 and stage 4 of the respiration, aerobic respiration. So this is actually glycolysis, the end of glycolysis. There is actually 2 more things to say. Few more things after this. This part will be over. After glycolysis, we need to study TCA cycle, which is the continuation of this process. Then we will study electron transport and forming ATP with the help of NADH2, FADH2 formed, release of carbon dioxide, and ATP synthesis. So just it is an easy process. If you understand the structure, it is an easy. First of all, you need to understand what you are studying. What I am studying in glycolysis. Glycolysis is the one of the process which is involved in the production. One of the process in a series of reaction which is used for the production of energy in the form of ATP needed in the cell. This is the end of the part 1. Actually, glycolysis is an anaerobic process, anaerobic metabolism. Why I am saying glycolysis is an anaerobic process? Because in glycolysis there is no need of oxygen. Glycolysis is happening in the absence of oxygen. So I can say that glycolysis is an anaerobic process. So after glycolysis, the glucose we consumed is being converted into 2 molecules of pyruvate. 1 glucose is converted into 2 molecules of pyruvate. 1 minute. One molecule of glucose is converted into 2 molecules of pyruvate. So actually glycolysis is an anaerobic process. Even in the presence or absence of oxygen, glycolysis will not change, because glycolysis always occur in the absence of oxygen. Because in this reaction, you can't see any oxygen coming into the scenario, coming into the scene. So I can say, or I can, in the sense, it is an anaerobic process. So what is the fate of pyruvate? The pyruvate formed by the process of glycolysis have certain fates. Fates means the formed pyruvate now is a substrate. it can be changed into different forms according to the availability of oxygen. In the absence of oxygen, in the presence of oxygen, it will be, this formed pyruvate can be entering into different forms. So the fate of pyruvate next is dependent upon the oxygen. In animals, if oxygen is plentiful, pyruvate will now undergo the next step, TCA, which means the pyruvate before entering TCA, we will say in the next part very easily, but just understand, right? The pyruvate before entering the next stage, which is just TCA, it will change into acetyl coenzyme and another molecule. Now the acetyl coenzyme can enter the TCA cycle. What is the condition? If the condition, this condition will only occur if the oxygen is plenty in the cell. If the oxygen is not plenty, if it is scarce in condition, what happens? The pyruvate will not form acetyl coenzyme. In animals, it will form lactate or lactic acid. We will draw the structure later in the next part. I'm just saying the basic idea, because I think the part 1 will be a very lengthy part. So I am planning to say it once more in the part 2. I'm just saying the basic idea right now. So pyruvate will be converted into acetyl coenzyme, and this acetyl coenzyme will be entering TCA cycle if the oxygen is plenty in a cell. And if the oxygen is scarce, very scarce, which means very low, what happens? In animals, it will form lactic acid. What happens in microorganism or anaerobic organism? You know what happens in anaerobic organism? Anaerobic organism always undergo their respiration without oxygen. That is the reason they are known as anaerobic. Some of the organisms are facultative and other are obligative, which means according to the environment they can be anaerobic organism and they can be aerobic organism. It is facultative. They can change their mode according to the absence and presence of oxygen. So in anaerobic organism, which are always undergoing anaerobic metabolism, their glycolytic pathway, what happens? They undergo glycolysis, pyruvate is formed, and pyruvate is converted into acetaldehyde, which then converted into ethanol. This is the condition. And this process is known as fermentation, because ethanol was earlier and now is used. Now also it is used in the production of, it is an alcohol. So microorganism which were used using this process is known as anaerobic metabolic process, which is fermentation. Production of ethanol. Carbon dioxide is also released in the case of yeast. Okay. This is the condition. Don't worry, we will be dealing this thing very deeply with structures in the next part. How, what is the fate of pyruvic acid or pyruvate in the presence and absence of oxygen. So in human beings, if it occur in the absence of oxygen, the pyruvate will form lactic acid. You know, if a cell needs immediate immediate energy, it will undergo anaerobic respiration. If a cell need immediate energy, which means consider an athlete is running, sprinting, is doing a sprint for 1 kilometer, 2 kilometer, 3 kilometer. What happens? For him, he need high amount of energy within a short period of time. So, and the oxygen availability is also very low in the cell. So, what happens? The cell undergo anaerobic respiration, which means glycolysis will be forming pyruvate. This pyruvate will be forming lactic acid. This lactic acid will be moving to the blood, and through the blood it will enter the liver cell. In the liver, pyruvate will be lactate will be converted back into pyruvate, and in the liver the pyruvate forms glucose by the process of gluconeogenesis, which is actually the reverse process of glycolysis, which means from pyruvate glucose is formed. This occurs in liver, and from the liver glucose enter the blood and it enters to the muscle cells or the whichever cells needed, and it undergo glycolysis. Thus, more energy is produced within a short period of time in the absence of oxygen. So, lactic acid will be accumulated. That is the reason for cramps. A diagram I have shown here. That is the reason for muscle cramp due to the accumulation of lactic acid in the muscle cells. Okay. So, powerhouse of the cell is known as mitochondria. Energy currency of a cell is known as ATP because they have a main function. Look, this is the overall idea of aerobic or respiration. This is the overall idea of aerobic respiration. Aerobic respiration. If it is in anaerobic, glucose to pyruvate will be forming lactic acid. It will not undergo these steps because if it is converted into acetyl coenzyme, it should enter TCA cycle. If it enter TCA cycle, it needs oxygen. So if oxygen is not available, this is a waste process. So if oxygen is available, it is according to the availability of oxygen, pyruvate will be converted to either acetyl coenzyme or lactic acid. I think you got an idea. Actually, it is little bit confusing. I will say it once more. The next part, from pyruvate to the fate of pyruvate. We need to understand we eat food, right? Food contains many compounds like protein, fats, vitamin, carbohydrates. So carbohydrates enter, mainly glucose enter the glycolytic pathway. There are other carbohydrate molecules also like galactose, lactose, mannose. They also have their own metabolism through which they will be changing and they will be entering into this pathway. For example, galactose is an epimer of glucose. They can also be metabolized. They also enter the glycolytic pathway. How they enter glycolytic pathway? Galactose will form galactose 6-phosphate and then it will be converted into glucose 6-phosphate. Now it can enter into the glycolytic pathway. How? Look, if galactose is converted into glucose 6-phosphate, it can enter in this step. Okay, so it will undergo glycolysis. We will discuss. Actually, it is not needed for according to NCERT, but the other sugar molecules also enter in this manner, like mannose 6-phosphate converted into fructose 6-phosphate, then fructose 6-phosphate enter into glycolytic pathway. Amino acids can also be formed. Some amino acids like alanine can be converted into pyruvic acid. So pyruvate is formed. This is the condition. I think you are getting a basic idea. We will go to the advanced in the coming lectures. And you know, ATP, glucovita. Look, in this product, glucovita, it contains glucose in a powdered form. So you know, glucose, glucovita powder, glucose powder, banana, which means these food substance have a ready-made glucose available in it. Banana is a fruit which contains ready-made glucose within it. So if we consume banana or glucose powder, the body can directly take the glucose and undergo glycolysis. It does not need any further process of converting the food because these are simpler molecule. If we eat food, actually carbohydrate digestion occur in the mouth, protein digestion occur in our stomach. So if the carbohydrate or the glucose is readily available, the cell can directly use and directly absorb into the blood, and the cells can take glucose from the blood and undergo glycolysis and readily produce ATP. So these are the uses of these substances, these food compounds. There are much more to say. There are much more to study. There are much more to understand. In the coming lectures, we will be dealing it. This is the overall reaction we will discuss in the next part 2. So thank you. I think you got a basic idea about glycolysis, breakdown of breakdown of glucose. Okay, I hope you like my video. We will be seeing you soon in the part 2, where we will be discussing the fate of pyruvate and the TCS cycle. And part 3 will be the next. We will be discussing about electron transport and ATP production. So I hope you like my video. There are much more to say. I hope. Much love. Thank you. God bless you.